Viral infection is a serious threat to human health. Viruses adsorb cells by binding to specific receptors on host cells, and rely on their own specific proteases for protein processing and nucleic acid replication. After viral infection, it generally goes through multiple stages such as adsorption, penetration, shedding, nucleic acid replication, transcription translation, packaging, etc. Preventing any of these processes can prevent virus replication. The most effective antiviral drugs should act on the two stages of virus adsorption and nucleic acid replication. Therefore, peptides that bind to host cell receptors or active sites, such as viral proteases, can be screened from peptide libraries for antiviral treatment. There are currently multiple antiviral drugs available in clinical practice, but most viral infections still cannot be completely cured. Scientists first discovered that peptides can resist microbial infections in the innate immune system. In recent years, a large number of antimicrobial and antiviral peptides have been discovered in animals, plants, and microorganisms. Most common antimicrobial peptides have broad-spectrum antibacterial activity, exerting their antibacterial effect by disrupting bacterial cell membranes and causing leakage of contents. In addition, peptides can also exert antiviral effects by inhibiting virus invasion, virus protein synthesis, and enhancing host immune function, providing a new source for the development of antiviral drugs. Antiviral peptides are an important part of current antiviral research strategies, which have demonstrated unique mechanisms of action and promising clinical prospects. Currently, well-known antiviral peptide drugs include the HIV fusion inhibitor enfuviride and the anti-coronavirus peptides HR1P and HR2P. Among them, the anti-coronavirus peptides HR1P and HR2P are the main products of Qiangyao Biotechnology. If you have any related needs, please feel free to contact us at any time.